Hello, and welcome to our series, What to Look For, where we talk about specific considerations for investigating foodborne illness outbreaks. In this video, we will discuss specific considerations for investigating Clostridium perfringens outbreaks. To start, let's go over some of the key characteristics of Clostridium perfringens, which we will refer to as the abbreviated C. perfringens for the remainder of this presentation. Firstly, C. perfringens is a spore-forming bacterium. And what this means is that when metabolically active or vegetative C. perfringens cells that are able to grow are exposed to unfavorable growth conditions, for example, conditions with limited nutrient access, low moisture, or non-optimal temperature, they can form internal structures called endospores, which are metabolically inactive. Importantly, these endospores are highly resistant to many stressors that would otherwise be lethal to the vegetative C. perfringens cells, such as exposure to high heat, drying, radiation, and certain disinfectants and sanitizers. Therefore, if a C. perfringens cell that has formed an endospore is exposed to one of these stressors, such as a heat treatment, then the vegetative cell will be lysed, allowing the endospore to exit the vegetative cell. And this structure, which is now called a C. perfringens spore when it is outside the vegetative cell, can remain dormant and withstand many of the same harsh environmental situations that the C. perfringens endospore can. Now, if this spore is later exposed to favorable growth conditions, for example, conditions with improved access to nutrients, high moisture, or temperatures that are optimal for C. perfringens growth, then it can germinate. This ultimately allows C. perfringens to transition back to a metabolically active state where it can once again grow. And so ultimately, what this means in the context of food is that C. perfringens cannot be killed or inactivated in food by most thermal treatments such as cook steps or pasteurization. Although it should be noted that some more extreme heat treatments, such as canning at temperatures exceeding 240 to 250 Fahrenheit for 20 to 100 minutes, can effectively inactivate C. perfringens spores. So, for example, if raw ingredients such as raw beef are contaminated with C. perfringens, then C. perfringens spores can still potentially be present in a cooked food product after a cook step is applied, even if the cooked food reaches an appropriate internal temperature. And furthermore, if that cooked food product is later exposed to temperature conditions that can support C. perfringens growth, then this can result in further outgrowth of C. perfringens. For example, C. perfringens can potentially grow at temperatures as low as 41 Fahrenheit in some cases. Thus, if a cooked stew is kept at this temperature for extended amounts of time, C. perfringens could grow to high numbers. In addition to being a spore-forming bacterium, C. perfringens can also produce toxins that cause foodborne illness in humans. These toxins can only be formed under favorable growth conditions that allow C. perfringens cells to grow. And more specifically, C. perfringens is only able to produce these toxins when it is growing within the human body. With respect to food, this means that if C. perfringens spores or C. perfringens vegetative cells are present at high levels on foods, then consumption of that food can lead to toxin production in the human body, which can result in foodborne illness. This particular type of foodborne illness is known as foodborne toxico-infection, which is defined as a foodborne illness that is caused by consuming a pathogen that proceeds to produce toxins in the human body after its ingestion. C. perfringens causes approximately 1 million cases of foodborne illness in the U.S. each year, and these cases are primarily associated with foodborne toxico-infection. Common symptoms of C. perfringens illness include abdominal cramps and diarrhea. In general, C. perfringens has an incubation period ranging from 8 to 16 hours, and the illness is typically self-limiting, with symptoms of C. perfringens associated foodborne illness resolving within 24 hours. However, symptoms can last between 1 to 2 weeks for select high-risk populations, such as infants and the elderly. Common local establishments where Clostridium perfringens outbreaks can occur include restaurants or other eateries, long-term care facilities, schools, community centers, and penitentiary facilities, just to name a few. When investigating C. perfringens outbreaks linked to local establishments, environmental health investigators should focus their efforts primarily on evaluating whether improper food holding or handling practices could have contributed to the outbreak. More specifically, when investigating C. perfringens outbreaks, efforts should be focused on evaluating practices that could allow for C. perfringens to grow in foods that require time and temperature control for safety, or abbreviated TCS foods. This is particularly important given that for C. perfringens, it is the toxins produced by C. perfringens, rather than the organism itself, that cause foodborne illness. TCS foods have specific characteristics that make them higher risk for growth of pathogens such as C. perfringens. For example, an abundance of nutrients, high moisture content, and low acidity or neutral pH. Some examples of TCS foods include meat, poultry, seafood, eggs, various dairy products, and fresh-cut fruits and vegetables. Some key practices that can allow for C. perfringens to grow in TCS foods that should be evaluated here include improper hot, cold, and room temperature holding practices, as well as improper cooling and reheating practices. When conducting outbreak investigations linked to C. perfringens, environmental health investigators should also identify whether any hot, cold, and room temperature holding practices or cooling or reheating practices used in the establishment were not performed correctly and failed to control C. perfringens growth on TCS foods. The goal here is to restrict the amount of time that TCS foods are exposed to temperatures within the temperature danger zone, which represents temperatures between 41 and 135 or 140 Fahrenheit, as these temperatures can potentially support the growth of disease-causing microorganisms such as C. perfringens. For proper hot holding practices, cooked or reheated TCS foods should be held at temperatures at or above 135 Fahrenheit to prevent C. perfringens growth. This can be done by placing cooked or reheated food on preheated tables, chafing dishes, or in slow cookers while serving, or by transporting food to different locations in the establishment via insulated hot food boxes. For proper cold holding practices, refrigerated TCS foods should be held at temperatures at or below 41 Fahrenheit to prevent C. perfringens growth. This can be done by storing foods in refrigeration units, walk-in coolers, or freezing units for long-term storage or thawing, or in containers with ice for short-term storage or serving. In general, establishments should limit the amount of times that TCS foods are held at temperatures within the temperature danger zone, which includes allowing foods to sit at room temperature. Specifically, TCS foods should be held for no more than 2 hours at room temperature before serving. Additionally, in cases where the ambient temperature in the establishment exceeds 90 Fahrenheit, TCS foods should be held at room temperature for no more than 1 hour before serving. It is important to ensure that TCS foods pass through the temperature danger zone as quickly as possible, and thus appropriate time and temperature controls should be applied when cooling foods. For example, TCS foods subjected to a cook step should be cooled to 70 Fahrenheit or below within 2 hours, and further completely cooled to 41 Fahrenheit or below within 6 hours. In another example, TCS foods such as a chopped tomato salad that are prepared from non-TCS food ingredients held at room temperature, such as whole uncut tomatoes, should be cooled to 41 Fahrenheit or below within 4 hours. Similarly, when reheating TCS foods that were previously cold held, for example in refrigeration units, it is important to ensure that the food is reheated to an internal temperature of 165 Fahrenheit for at least 15 seconds within 2 hours after it was removed from cold holding conditions. On the other hand, if food has potentially been subjected to temperature abuse, for example being left out on the counter overnight, then reheating of food should not be performed, and instead the food should be discarded. This is because both C. perfringens endospores and spores are fairly heat resistant, so reheating is not an effective process for inactivating C. perfringens on foods. Environmental health investigators can utilize multiple approaches to evaluate whether any improper food preparation and storage practices, such as improper hot, cold, or room temperature holding practices, or improper cooling or reheating practices, could have contributed to a C. perfringens outbreak. These can include reviewing relevant food-associated records, conducting interviews with upper management and food handlers at the establishment, carefully observing food holding and handling practices during site visits, as well as collecting food product or ingredient samples from the establishment and testing them for the presence of C. perfringens. Key records from the establishment that environmental health investigators should aim to review can include 1. Standard operating procedures such as SOPs or recipes, and 2. Documents where monitoring and verification activities are recorded. In particular, when reviewing SOPs, investigators should pay careful attention to hot and cold holding procedures, as well as reheating and cooling procedures, to make sure they are appropriate and in line with time and temperature controls that can control the growth of C. perfringens on food. Additionally, through reviewing, monitoring, and verification records, the investigator can ensure that SOPs are consistently being followed. For example, investigators should review temperature check logs to identify the frequency in which they are being performed during hot holding on a steam table or during cold holding on ice, as well as logs that show how often thermometers are calibrated and what do calibration records say about the accuracy of the thermometers. Other documents that might be particularly important to review may include checking if cooling and reheating logs show that foods were able to reach their endpoint temperature in an appropriate amount of time. For example, in a 2008 C. perfringens outbreak associated with a casserole prepared at a county jail in Wisconsin, during a review of records, environmental health investigators identified that the temperatures of food during hot holding were not routinely obtained and or recorded consistently, and the establishment did not have appropriate documentation of cooling step temperatures for cooked beef and macaroni, both of which represented ingredients that were used to prepare the casserole. To supplement this review of records, investigators should also conduct interviews with both management and food handlers at the establishment and ask questions about 1. How SOPs for various food preparation and storage practices are enforced, 2. Any potential inconsistencies in SOPs and monitoring or verification records, as well as other questions about 3. Food thermometer calibration and maintenance. Whenever possible, investigators should observe or ask food handlers to demonstrate certain tasks, such as cooling and reheating practices. In addition, when conducting site visits at the establishment, environmental health investigators should closely observe how food is routinely prepared and stored. During these observations, one key thing to take note of is how procedures for hot and cold holding are implemented in the establishment. For example, are steam tables preheated prior to hot holding? Is there sufficient space in refrigeration units or walk-in coolers between food products to allow for proper cold holding? And how frequently are food handlers checking the temperature of foods when serving foods that are temporarily being hot held or cold held? Additionally, environmental health investigators should examine how procedures for reheating and cooling are being implemented in the establishment. For example, are foods being rotated or stirred during the reheating process to ensure even distribution of heat throughout all parts of the food? And is the appropriate equipment being used to facilitate cooling? For instance, for foods such as soups and stews in large containers, using ice or cooling paddles may accelerate the cooling process. Finally, environmental health investigators may want to collect select food product or ingredient samples from the establishment and test them for the presence of C. perfringens. One key thing to consider when collecting food product samples is ensuring that all collected samples are transported to the lab appropriately. More specifically, it is important to ensure that food product samples collected for C. perfringens testing are stored at refrigeration temperatures or lower during transport to the lab. This is particularly important as, according to the CDC's guidelines for confirming the cause of foodborne disease outbreaks, in order for a food product to be implicated as the cause of a C. perfringens outbreak, the food product needs to contain more than 100,000 or 10 to the 5th organisms per gram of food. thus ensuring that food samples destined for C. perfringens testing are subjected to refrigerated temperature conditions or lower during transport is imperative for preventing possible growth of C. perfringens. In summary, in this presentation, we have covered some of the specific characteristics of C. perfringens, as well as key areas that are relevant for environmental health investigators to consider when investigating C. perfringens outbreaks linked to local establishments. In addition to C. perfringens, check out our other videos that cover key considerations for investigating foodborne illness outbreaks linked to Bacillus cereus, Staphylococcus aureus, norovirus, hepatitis A virus, Salmonella, and Campylobacter.